Good afternoon. My name is Kane Akai. I'm one of the senior healthcare analysts here at Chardan, and it's my pleasure to introduce our next guest from Life Edit, Senior VP, Corporate Development and Operations, Dr. Claire Murray. As a reminder to attendees, if you'd like to ask a question of our speaker, please type it into the question box under the video player, and we will try to ask them on your behalf. Claire, first of all, welcome. Thank you so much for being with us today. And to start out, could I ask you to provide a couple minutes of introductory comments about Life Edit for audience members who are new to the story?
Yes, and thank you, Kay, for having me. It's a pleasure to be here. So yes, Life Edit is a gene editing company based in Durham, North Carolina. We're a wholly owned subsidiary of Elevate Bio, who I believe is also a part of this program, maybe later in the
Tomorrow,
day.
yeah.
Tomorrow, even better. And so we're a company, we're building a broad gene editing platform that really is built on the foundation of a set of nucleases that we identified from a proprietary collection a number of years ago. And we've been able to use that foundation to develop approaches to double-stranded breaks, base editing, and RT editing. And we're also now bringing in the delivery components we're going to need to be able to get those gene editing systems to the tissues and organs that we're trying to get them to.
Well, great. So I think our audience is pretty familiar with gene editing. You know, obviously everybody's aware of the work by Dr. Doudna, who's presented at some of our conferences in the past. So, Claire, what are some of the specific problems associated with gene editing that you're trying to solve with your platform?
I think a couple of things. So, we want to build this very broad platform so we can facilitate being able to really undertake any edit anywhere. And so we want to set up a world where you don't have to ask the question, what disease or disorder can I address with the tools I currently have in my toolbox, but rather say, what disease or disorder do I want to address, treat, cure, and which of the tools that I have available are there and I can bring to bear on the particular indication. And so we think that to be able to do that, you're going to need a broad base of nucleases with a wide diversity of PAMs. We think you're going to need different editing modalities to be able to make the edits you want to make. And we think that both non-viral and viral delivery systems are going to be important to be able to deliver on ultimately that vision. And so that's the company we're building.
And again, I think you mentioned PAM. I think our audience understands that, that's critical to directing where the edits will occur. And, the canonical PAM of the SpCas9 is any nucleobase followed by two guanine nucleobases. So can you tell us about the PAM diversity of your RGN library?
Yes, yeah. So I mentioned earlier, we had an opportunity to mine a proprietary microbial database, really, or collection, to look for these nucleases. And when we did that originally, we recognized that within that collection, there was a broad number of CRISPR systems with this very, very broad PAM diversity. We've augmented that actually subsequently through metagenomics mining of various databases. And what we decided to do was to focus on identifying nucleases that would really give us orthogonal coverage of genomic DNA. And so we have a number of lead RNA-guided nucleases at this point that get us an ability to go after a lot of the genomic DNA. We continue to think about different ways we need to augment that collection with other CRISPR systems. And then this PAM diversity is particularly important if you're going to do base editing, because you need to be able to be within a certain region of the PAM to be able to do the base edit. And so we've leveraged that a lot with our base editing platform. And what we've also done is made all our systems very modular. So you can choose the nuclease and the PAM that you want to use, and then look at the deaminase you need for the base editing or the other components you need for RT editing. But we've found it to be really facilitating for base editing and really important if you want to get to, if you want to be able to do edits in a number of different places in the DNA.
Okay, great. So, I think in looking at the company, it seems to be a combination of both the technologies you're developing that are available to other drug developers, but you also have your own pipeline of proprietary products, the first one being a program targeting Huntington's disease. So tell us, why does Huntington's represent an appropriate initial target to achieve proof of concept for your design platform?
Yes. So I think first and foremost, Huntington's disease is a monogenic disease. And so it clearly fits into the kind of landscape of indications that are ideal for gene editing. So for those who aren't as familiar with it, this is caused by a single mutation in the Huntington gene. It leads to this CAG trinucleotide expansion. That results in aggregates of the mutant Huntington that ultimately leads to progressive neurodegeneration. There's a really significant unmet need in Huntington's disease. Currently, there's no treatment, there's no cure. And so we felt, between the fact that it was monogenic and the fact that there's really no current treatment, that it was really a great indication for our gene editing platform. There's another detail within Huntington's disease, which is in order to treat it, you want to be able to selectively edit the mutant Huntington and leave the wild type Huntington free of edits. And so using our collection of RGNs and leveraging our PAM diversity, we've been able to separately target both the T and the C alleles of a PAM altering SNP. And we've been able to do that in an allele selective fashion. And so we think that we've got an approach that is quite exciting in the Huntington's disease field. To complete this is an AAV. This will be delivered intrastriatally with AAV5. And so we're excited as we're bringing this asset forward.
Okay, great. Yeah, so clearly, I think Huntington's could be a great opportunity if somebody's able to crack the code there, and obviously the allele specificity is just a super attractive feature. Now, you did mention that you will be delivering using AAV. I think as we talk to our gene editing companies, they're kind of, some use AAV, others use LNP. So why for this indication and your payload does AAV make more sense?
So I think first and foremost, we're talking about delivering to the CNS and specifically to the brain, and there is plenty of precedence for the delivery of AAV to those to the CNS and the brain. So it was a way to keep at least the delivery system consistent with precedence. And then in addition to that, we didn't need to think about the size of our payload. One of the features of our gene editing systems or our nucleases is that they are smaller than SPCas9. And so we're really able to package our nuclease, the regulatory elements, the guides, and everything into a single AAV. So we just felt, from a product perspective, it was the right decision to go with a viral delivery system. Okay,
great. So can you describe some of the key preclinical data you have to date for this candidate?
Yeah, so we're currently wrapping up a dose finding study in NHPs, and we anticipate having that data in the next few months. Prior to that, we had done in vivo work in the BACHD mouse, where we're really showing both the efficacy of the gene editing composition as well as its allele selectivity. And so as we get the data from the dose sighting study, that'll be the data that will allow us to start deciding how to move forward towards the clinic.
So I guess that would suggest that perhaps the next step is IND enabling talks.
So I think what we want to do at this point is get the data from the dose sighting study and look at where we are, and we'll start to make the decisions from there.
Okay, great. And so, you're wholly owned by Elevate. So talk about, how you're set up with them from a manufacturing perspective.
Yeah, so it's really great that we're part of the Elevate Bio ecosystem. So we can focus on the gene editing, preclinical development. Elevate Bio has significant manufacturing expertise in cell therapies and in vector. And so we've been able to leverage that expertise. The team at Elevate Bio has done the process development, analytical development for our asset. They manufactured the material that we used in the dose sighting study, and it's really been a great relationship between the teams. We've been able to bring all the gene editing expertise. They've been able to bring all the manufacturing expertise, and so we look forward to continuing to drive that forward together.
And having, I guess, started with a CNS target, if one were to think about what you might do next in your pipeline, is CNS going to be the primary focus area?
I'm not sure if I would describe it as a primary focus area. What I would say is we've built a lot of expertise in CNS delivery through the Huntington's disease program. We think about how we're going to leverage that for other indications. But we've also been building experience delivering to the liver. That we recently, or Elevate Bio recently acquired an LNP platform that we've been able to start using, and we're starting to generate data showing the delivery of the gene editing systems. We think about other organs such as the eye. So I think we're not going to just be one and done in the CNS. We definitely want to leverage what we've built there, but we also see that there are other organs and tissues that we would be excited to get to.
Okay, so again, as a provider of technology to other companies, you do have two announced collaboration agreements, one with Novo Nordisk and the other with Moderna. What can you tell us about these agreements?
Yeah, so what I can say is the collaborations are going really, really well. We're having a lot of fun with both partners. Both partners came to us because they were excited by the breadth of the platform, and in both cases, some of the depth and the robustness of the base editing platform. We are working closely with them. Obviously, we bring the gene editing expertise. They bring the drug development, clinical development expertise. So everything's going well at this point. I think we're just over a year into the Moderna collaboration, just around a year into the Novo collaboration. So we're having a lot of fun.
So beyond financial considerations, what is Life Edit learning from working with these collaborators?
Yeah, that's a good question. I mean, I think we're learning every day. With the collaborators, they're pushing us to think about how we can accelerate development of various aspects of our platform. They continue to push to understand, how we can not only develop the gene editing composition, understand the on-target effects, but also thinking about what does the off-target profile look like? And so really building all the depth and breadth of tools that you need to be able to move these forward. And then I think, with a companies like Novo Nordisk and Moderna, they understand what a product's going to look like to be successful in the clinic. And so they're really infusing these early programs with that depth of knowledge. And we think that that's going to make for very successful products down the line.
Okay, so I guess in a bigger picture, is there a strategic goal in mind in terms of maybe the appropriate balance between, again, being a provider of technology to others versus developing your own proprietary candidates?
Yes. So really, our strategy is to identify and develop partnerships or collaborations with partners who have an expertise or a technology or are bringing, something to the table that will allow us to leverage this platform to its fullest potential. So we don't really see that as competing with internal programs. We see it as an opportunity to be able to really use our gene editing platform as broadly as possible and build a pipeline that has, assets that are wholly owned by us, maybe partially owned with future partners, and then wholly owned by our partners. So we think that that's the best of both worlds. And we think that we can work in both those environments, both on our own platform and our own therapeutics, as well as delivering to our partners.
Okay, so when we're thinking about maybe inbound interest on the partner side, would you say that more of that is because of what you described for the first two, more of that is focused on where the field might be going in terms of either base editing or prime editing versus more traditional or original gene editing, double-stranded break?
That's a good question. I think when we think about partnering, we're really thinking about what's actually needed for the therapeutic in hand. So I do think partners come to us because we've got this broad modular platform and where they can get access to, cutting edge technologies. But also, just as with the Huntington's disease program, there's a place for double-stranded breaks and nuclease activity. And so I think we can partner in all those spaces and successfully drive things to the clinic.
Okay. So in terms of what the company is focused on currently in terms of advancing the science and capabilities of your platform, what would those things be?
Yeah, so I think we continue to refine the base editing. We continue to build the RT editing. We're thinking a lot about the ability to insert larger payloads, both from an editing perspective and from a payload delivery perspective. And then I think just like everybody else, trying to really understand what are we going to need from a delivery perspective. With the Huntington's disease program, we've used AAV. We've been looking at other viral platforms and where those might intersect with our gene editing. And then we have a team that's really digging into the LNP and non-viral delivery. And so I would expect over the next few months, there will be data showing our gene editing systems in conjunction with our LNPs being delivered to both the liver and potentially non-liver tissue.
Okay. And in terms of the reverse transcriptase for prime editing, obviously, kind of a more novel idea, slightly different guide, RNA, the nickase, Cas9. What are you doing to enhance the characteristics of those elements for RT?
Yes, we're doing a lot of different things in this space. But I think the kind of one area where we're focused is trying to make it as modular as possible so that we can use it with all our different nucleases. This work's underway now, and as I say, we're hoping that we'll be able to start presenting robust data on that, later this year.
Okay. And in terms of publications or presentations at scientific conferences, is there stuff you have scheduled for the balance of this year that we should be looking forward to?
Yeah, so a lot of what we're doing right now, we're really spending time filling out our IP strategy, making sure we're filing all the patents we need to file. We do have our scientists going to a number of conferences over the next few months. So you should expect to see us at kind of all the big conferences. We do have a team going to ASGCT. We don't have posters there this time, but others from Elevate will be presenting.
Okay. And I guess in terms of partnerships, obviously, you're not stopping at two, but
Yeah.
again, how far do you want to take, this aspect of your business?
Yeah, I think it's a fundamental part of our business. It's not something that we're just doing a little bit on the side or doing it lightly. We have a number of different partnership structures that we set up, from non-exclusive licenses, getting access to our gene editing systems to enable cell therapies. Those are pretty straightforward licenses, and we envisage that we'll be providing, a fair number of those, all the way through to deeper collaborations like we have with Moderna and Novo. I think there's plenty of room for additional collaborations at that level. And then in addition, as we're developing some of these assets internally, looking to see if there are opportunities to partner those for the later stages of preclinical and early clinical development. So we think there's we think there's a massive landscape for forming collaborations and partnerships. And as part of Elevate Bio, that's a fundamental part of our business.
Okay. A question on the chat. Where are you in terms of discussion with regulatory agents for Huntington's? How do you know you're on the right track without any kind of roadblocks to entering the clinic once you complete some of the traditional tasks, like IND enabling?
So we will be getting regulatory feedback on the asset later in the year. To this point, we've been working closely with CHDI and other groups that have been providing kind of a roadmap and guidance for taking this forward. But absolutely, we are looking forward to sitting down with regulators later in the year.
Okay. Well, maybe just a final question and maybe a way to summarize all the things we've just talked about. But what do people really need to understand and appreciate about what you're doing at Life Edit?
Yeah, I think there are three big things. Number one, we've built a broad next generation gene editing platform that we are excited to partner with others. And we sit under the Elevate Bio ecosystem. And so you can work with us from very early discovery all the way through to manufacture of either your cell therapy or your in vivo gene therapy. And we can do that with a light touch or with as deep a touch as a partner needs. So that's it.
Okay, well, very good. We've reached the end of our time here, but I want to thank you so much for joining us today, Claire.
You too. Thank you. I appreciate it.
Great.